Hello and welcome everyone. Thanks for joining us today for this Lab Medica webinar, Enhancing Efficiency in Laboratory Medicine, a focus on quality control procedures. My name is Kate and I'll be your moderator for today's presentation. Today's webinar is presented by Lab Medica and sponsored by LGC. Throughout the presentation, please feel free to submit any questions that you may have. You can do so by simply typing in your question at the bottom of the chat tab on the right side of the screen. And we will respond to as many of your questions as we can during the question and answer session, which will be after the presentation, so therefore towards the end. And with that, I'm delighted to introduce our speaker today, Oswald Sonntag. Oswald Sonntag is an independent consultant in the field of laboratory medicine. Due to his long-term experiences with quality control management, he is a well-known expert in this field. He was involved in the early days of setting the first steps of the famous Relibec in Germany. His experience is broad, as he has worked in both university labs and in different diagnostic companies. I will now pass it over to Oswald to begin his presentation.
Thank you, everybody, for joining me, and also a special thank for LGC Technopass for sponsoring this event and for allowing me to give you a presentation on enhancing efficiency in laboratory medicine. Today, a focus on quality control procedures. Quality is achieved by doing the right thing right, the first and every time. And in our definition or in our understanding, the right thing is the requirement or expectation of the physician or patient. And this quality contributes definitely to patient safety and on efficiency. And today, as I said, we will talk about efficiency. The definition of efficiency is the often measurable ability to avoid wasting materials, energy, efforts, money, and time while performing a task. In a more general sense, it is the ability to do things well, successfully, and without waste. Definition of efficiency in the view of the laboratory medicine. Efficiency in laboratory medicine is vital for high quality patient care and as well for medical research. It involves obtaining accurate, precise, and prompt results while minimizing resource consumption and encompassing time, materials, and personnel. Our focus today is especially the efficiency with the use of quality control material. And a very important part in our field is to ensuring patient safety. So efficient laboratory processes have a direct impact on patient safety by ensuring timely and accurate results. Reliable results are crucial for informed medical decisions, and any delay or inaccuracy can pose risk to patient well-being. Therefore, we have to focus always on quality. And what is the quality control procedure in our field? Quality control procedures in our field refer to systematic measures and protocols implemented to ensure the accuracy, reliability, and consistency of test results, thereby maintaining the high standards of quality in patient care and research. So a typical graph which we are using in the field of laboratory medicine is the so-called Levi-Jennings chart, which you see here on this slide. And you're probably familiar with this graph. We have the mean value, and we have the 2 and 3 SD, standard deviation ranges, um, which usually should be used as the criterion or the range or limitations which we use for running a quality control material. So typically, we are measuring each day or each run 2 or 3 different levels of quality control material. This have to be analyzed prior to the measurement of patient samples. The QC material plays a crucial role as it mimics a patient's sample, but it has predetermined values used to verify whether the analytical process falls within specified limits or not. It is essential for ensuring reliable patient results. Typically for my presentations, I'm not that much in favor using abbreviations. And if I use an abbreviation, I try to give you the explanation for it. So if I talk about QC, that means quality control. The importance of efficiency in laboratory medicine. First hand is patient care. Efficient laboratory processes are essential for patient care. Timely and accurate test results are critical for diagnosing diseases, monitoring treatment effectiveness, and making informed medical decisions. Delays or inaccuracy in test results can lead to misdiagnosis or incorrect treatment, potentially harming patients. Cost effectiveness, efficiency can lead to cost savings. And at the end of my presentation, I will show you a real example of a study which was done showing how efficiency contributes to saving costs. Inefficient laboratory operations can result in excessive resource utilization, such as reagents, equipment, and personnel time. Streamlining processes and reducing waste can help laboratories operate more cost effectively. Importance of efficiency in the laboratory medicine touches also research and development. Efficiency is crucial in the field of research and development within laboratory medicine. Researchers rely also on accurate and timely data to conduct experiments, analyze results, and make scientific advancements. Efficient laboratory processes enable researchers to work more effectively and accelerate the pace of discoveries. Compliance and accreditation. Many laboratories are subject to regulations and accreditation requirements. So, for example, some labs are following the ISO 15189 requirements. Therefore, it's also important to look at efficiency from that point of view. Efficiency plays there a role in meeting these standards as well as maintaining records and documentation. Efficient processes ensure that laboratories are compliant with quality and safety standards. To achieve efficiency, you may use consolidated quality control material. But what is a key quality control material, which is called to be consolidated? The definition here is a consolidated quality control material is a sample that contains multiple analytes of interest. It serves as a material against which the results of various tests and measurements can be compared. This control material consolidates multiple quality control elements into one sample. The composition of such material is these materials are designed to mimic like a real patient sample and may contain a combination of analytes, for example, glucose, cholesterol, electrolytes, and other substances that are routinely measured in clinical or scientific testing. The composition depends on the specific tests the laboratory or quality control process needs to monitor. I have here a picture which shows you quite easily the difference between a consolidated material, which you see in the front of the picture, the three tubes there or three bottles there, and a non-consolidated material, which you see in the back of this picture, these 12 bottles. Now, to explain you the difference, it's quite easy. If you look at all these 12 bottles, these are non-consolidated material. And if you look at the same analytes which you want to measure from those 12 bottles, they are in the three bottles in front of this picture. So that's the consolidated material, which includes all the analytes which you usually may use and you buy different bottles with different components in it. Those consolidated material should be also labeled, so that makes the use quite easily. So the benefits of a consolidated quality control material: by consolidating multiple analytes into a single sample, laboratories and testing facilities can streamline their quality control procedures. This reduces the time and effort required to prepare and handle individual control materials for each analyte. This efficiency can lead to cost savings, increased in, and increased productivity. The benefits of a consolidated quality control material is also minimizing the risk of errors and contamination. Using such a material reduces the number of steps involved in quality control processes. Fewer steps means fewer opportunities for errors to occur and less risk of contamination. This can lead to more accurate and reliable results, which again are very crucial for the medical labs. Improve precision and reproducibility. Consolidated quality control materials provide consistent reference standards for multiple analytes, making it easier to assess the precision and producibility of testing equipment and methods. This helps ensure that measurements are consistent over time and between different instruments or operators, improving the overall quality of testing and research. Cost effective. Using a single control material for multiple analytes can be more cost effective than purchasing separate quality control materials for each analyte. Laboratories can save on procurement costs and also on storage space. Homogeneity. It should be highly homogeneous, meaning that all components and analytes which are usually uniform distributed in that material. This ensures that each portion of the material is clearly a representative of the whole, allowing for accurate and consistent quality control results. Stability. The material should have a high stability over time and also under various storage conditions. This stability ensures that the quality control material properties and analyte concentration remain consistent, enabling long-term use without significant changes. The matrix should closely resemble the matrix of real patient samples. For example, in clinical laboratories, the matrix should mimic like human serum or urine. This similarity ensures that the quality control material behaves like real samples when subject to testing procedures. multiple analytes. It should contain a range of analytes relevant to the specific tests or measurement being performed in the laboratory or quality control processes. It's important to cover a variety of analytes to assess the precision of multiple analytes simultaneously. Assigned values. Yeah, the analyte concentration within the quality control material should be accurately and precisely determined. These assigned values serve as a benchmark against which the results of routine testing are compared. The values must be established through rigorous testing methods. Ease of use. An ideal quality control material should be ease of use, allowing for convenient and efficient quality control procedures. This includes user-friendly packaging and clear instruction for implementation. Therefore, a liquid material is of preference. Compatibility. It should be compatible with the specific testing equipment and methods used in the laboratory. Compatibility ensures that the quality control material can be readily integrated into the quality control process without significant adjustment or adaptations. Yes, should have also a long shelf life. It should have a long shelf life to reduce the frequency of material replacement and minimizing the waste. This is particularly important also for cost effectiveness and operational efficiency. Documentation. The quality control material should come with comprehensive documentation that includes information about its properties, preparation, storage, handling, and the recommended usage. This documentation is crucial for maintaining quality control standards. Um, this is especially important to me to read the documentation, which are very often called instruction for use. They are including important information which you may use also in case of troubleshooting. So please read this documentation. Availability. It should be readily available and accessible, ensuring that laboratories and quality control facilities can procure it when needed. Current workflow. In many laboratory and quality control settings, the current workflow may involve the use of multiple individual quality control materials, each specific to a particular analyte. This process typically includes the following steps, which can be time-consuming and increasing the risk of errors. Material procurement. The laboratory staff needs to order, receive, and store various individual quality control materials for different analytes. This may involve tracking multiple suppliers, ensuring adequate inventory. Material preparation. For each analyte, separate quality control materials needed to be prepared. This involves measuring, diluting, and possibly reconstituting the materials to achieve the desired concentration or target values. This step is prone to errors in measurement and calculation. So these all is not necessary in a consolidated QC material labeling. Each individual quality control material must be properly labeled, indicating its analyte and concentration. Labeling errors can lead to incorrect usage. Testing. The individual quality control materials are used for testing involving multiple handling steps, reconstitution, and calibration of testing equipment for each analyte. Data recording. After testing, the results for each analyte must be recorded separately, increasing the chances of transcription errors. The recorded data for each analyte are analyzed individually, and corrective action are taken for each if necessary. Streamlined workflow. In contrast, a streamlined workflow utilized a consolidated quality control material significantly reduce handling steps and therefore associated risks. Material procurement, the laboratory only needs to procure and maintain one consolidated quality control material that covers multiple analytes. Material preparation, since the consolidated quality control material contains a variety of analytes, there is no need for separate preparation steps for individual quality control materials, reducing the potential for measurement and preparation errors. Barcoded QC products come ready to use in primary tubes that can be loaded directly onto the instrument, and in some cases can even be stored on the instrument for specific periods of time. Testing. The consolidated quality control material can be used as a single material for testing multiple analytes, simplifying the testing processes. Data recording, results obtained from the consolidated quality control material are recorded once for multiple analyses, minimizing there the risk of transcription errors. And the analysis involves a single quality control material which simplifies data interpretation and corrective actions. Now to show you the difference in handling with the two quality control materials which I talked about. So the non-consolidated quality control material on the left. On the right side, the consolidated quality control material. So for the non-consolidated, you have several bottles. As you can see, there's 12 bottles here. Each bottle needs to be opened. So you open the cap, and then with the pipette tip and the pipette, you have to transfer the QC material into a cup. Therefore, you have a risk of handling errors, but also carryover may happen and or mislabeling of the cups. If they are not done properly, errors will occur and problems will occur. Finally, also delaying the reporting of patient results. With the consolidated quality control material, if they come in those tubes, which you see on this picture, you just remove the cap. Done. You see, usually a picture tells you once often much more than a number and a lot of number of words or sentences. Avoiding sample mismatch. So with the comparison here, consolidated, they are in the front, the 3 bottles, the 12 bottles representing the same content as the 3 bottles in front, but they are non-consolidated, the 12 bottles. There's a risk of sample mismatch when using multiple quality control materials and especially vials. Consolidated quality control materials reduce the chance of QC sample mismatch. And if labels are directly on the tube, you can avoid any sample mismatch. Smoothing the quality control process. The challenges in quality control in the laboratory can be addressed through the use of consolidated control materials and automation. Quality control materials provide standardized reference points to verify the calibrated instruments and monitor performance, while automation enhances precision, consistency, and efficiency in the quality control process. These combined strategies contribute to ensuring the accuracy and reliability of laboratory test results and ultimately patient safety. Here is also the comparison between a lyophilized material or the use of a liquid quality control material. Lyophilized quality control material, very often known also as freeze-dried material, necessitates the laboratory professionals to reconstitute the sample by adding deionized, sterilized water, or sometimes also a special solution, a buffer, and mixing it before use. While this format may not be as user-friendly as liquid control, it offers distinct advantages. So I want to be fair if I'm comparing two different things. The increased stability of lyophilized control material extend its shelf life to nearly double the standard 2 years associated with liquid controls. However, the potential for reconstitution errors and its relative complexity make the majority of laboratories opt for the more convenient liquid controls. Using liquid quality control materials helps minimizing the risk of reconstitution errors and guards against contamination from low quality water. While liquid frozen controls need to be stored in freezers and thawed in a refrigerator prior to use, they are slightly less convenient compared to the ready-to-use liquid alternative. However, they eliminate the need for reconstitution, reducing the potential for associated errors. To maintain their frozen state during transportation and prevent thawing in transit, these controls are often shipped with dry ice, which can result in extra transportation costs. There are also two types of quality control material, which I called here assayed and unassayed. What's the difference? In an assayed quality control material, the target values and acceptable ranges are offered by the manufacturer of this quality control material. While unassayed material, it has to be analyzed by the laboratory to determine the target value and acceptable ranges. Crossover studies need to be run between the current and the new lot. So the action which you may have to take in order to use the unassayed quality control material is written down here. You have to calculate the mean and the standard deviation from data which have to be generated prior to the measurement of patient samples. So usually 10 to 20 days, depends on your local regulations, may be needed to measure the unassayed quality control material. So you have to plan about 10 to 20 days ahead of the use of such an unassayed material in order to get your mean and your limits. Important to note is that no data point should be excluded unless they are known to be a result of an operational error. Less crossover studies needed. A crossover study on quality control material in medical laboratories conducted during the introduction of a new lot, QC lot, entails a comprehensive comparison of the performance between the existing and the new batches. In a randomized order, laboratories employ both lots, exactly gathering data on accuracy and precision. Subsequently, statistical analysis employed to ascertain whether there are any significant differences, thus ensuring that the introduction of the new lot does not compromise the reliability of test results. So in short, crossover studies are required in quality control material lot changes, and these are time consuming. Each quality control material may have lot changes. So it's typically that if you have a lot of different bottles of quality control material with a lot changes, um, you have to do those crossover studies moreover. And in order to reduce this, my recommendation is to order a decent amount of quality control material for a long period of time. This gives you time and resource savings. Just to explain you also a little bit the lot-to-lot issues with the reagents, I found this paper from Franz Stroyenstein from the Netherlands. And here you can see on the left side the graph, the within day imprecision or precision. looks quite good in this example. Look at the day-to-day imprecision. You see there is a higher frequency and the imprecision is a bit higher. And then the impact of a calibration. So calibration is an adjustment of mm, the instrument and the cooperation between the instrument and the reagent. And you see here on the third line of this graph on the left side, the impact of the calibration. It may increase or decrease the values. And then if you have a reagent lot change, this has also an impact, which could be a heavy impact or not that heavy, depends on the differences of the composition of this reagent lot. And finally, at the end of the graph on the left side, you see the total variance. And if you compare this with a within day imprecision, yeah, you see the real difference. On the right side, that is also um, describing patient samples, measuring once per reagent lot. That's the figure A there. And you see the red dot, that's a patient sample, behaves similar to the QC sample. And each time the dotted line shows you the lot change of a reagent has an impact here. And in this paper, it's recommended to use patient samples more often together with the QC material to detect those uh, differences and shifts with using a new reagent lot. Now, at the beginning of my presentation, I told you that there was a study run in Spain, and I'm happy to share you a little bit the outcome of this study in regard to the efficiency, the topic of today. So a study conducted at the Ramon y Cajal hospital in Madrid, Spain, highlighted the consequences of laboratory inefficiency, such as delays or errors, underscores the critical link between streamlined laboratory operation and enhanced patient care. This connection emphasized the importance of investing in and maintaining efficient laboratory practices to uphold the highest standard of patient safety in healthcare. So this paper was published in the Journal of Laboratory Medicine in the end of 2022, and it's called Consolidation of Quality Control Materials Improves Patient Safety and Supports Sustainability. I took a table out of this publication and just showing you the results of the study which the lab at the hospital in Madrid found in this study here. So I blanked out the name of the manufacturer of the QC material. Here you just see non-consolidated and consolidated as the name of the QC material. Now, if we compare the metric which they use, the cold storage space requirement, and you see they found a saving of 90% for the immunoassay controls, and for the clinical chemistry controls 50%. The QC handling time, 99% was saving time if they use consolidated quality control material in the immunoassays, and nearly 100% for the clinical chemistry. The dead volume, so that's the volume which is left over after doing the measurement of the quality control sample on the instrument. And here you see a saving of 79% using consolidated quality control material comparison to non-consolidated immunoassays, and 100% in clinical chemistry. Waste from vials, there you see the savings of 50% in immunoassays, and even in clinical chemistry a saving of 40%. a waste of cups and pipette tips, because with the consolidated material you can use the tubes directly. You don't create waste due to cups and pipette tips. So in both cases, 100%. So this study shows clearly the advantage of using a consolidated quality control material by delivering the same performance. To show you a little bit more the waste they are produced and therefore compared. You see, if I have a consolidated quality control material, here these 3 bottles, I need to have these 12 bottles for non-consolidated quality control material. If I run the immunoassays they have tested in this study in Madrid. For clinical chemistry, you have 3 bottles only, or 3 tubes only for the consolidated, while for clinical chemistry, you have 5 bottles here. Now look at the packages. The waste you're producing with non-consolidated material. Here, as an example for the immunoassays, you see it on the left side, the waste which was produced in this study, and on the right side, in comparison, uh, quite dramatically less waste produced with consolidated quality control material. Now showing you also the consumables and also the waste produced with the consumables. You see the consumables produced in the non-consolidated quality control material on the left side. On the right side, only the few tubes which are produced in this study, just to give you a balance between the 2 different QC materials. So I'm nearly at the end of my presentation, and like to summarize a little bit what I talked about today. So the efficiency goals is that you have to be accurate, precise, and prompt results. With the use of a consolidated material, you minimize the resources you used, the time, materials, and also the personnel. So this is given definitely by using high consolidated quality control material, and this was supported by the study run in Madrid. The benefits: reduced handling steps, therefore avoiding sample mismatch, cuts overall expenditures, enhance efficiency, and reduce storages needed. Reduce crossover reliance, larger QC material orders, consistent quality, and fewer transitions. Balancing efficiency and quality, optimal lab practice consolidates quality control material, maintains equilibrium. Finally, patient safety is critical, and therefore you have to ensure result reliability. And again, as the study from Madrid shows us, that is given by the use of highly consolidated quality control material. Conclusion: consolidated quality control material integration boosts efficiency, patient safety in diagnostics. And my very last slide, you see here juggling with too much quality control materials, just to show you a little bit in a nicely format, the white packages on the left side, on the right side, the numbers of non-consolidated quality control material. So thank you very much for listening to me. I have now to do a technical step to move you back.
Thank you very much, Oswald, for your informative presentation there. We will now start the question and answers portion of the webinar. But just before we begin, I'd like to remind our audience to feel free to continue asking questions by just typing your question in at the bottom of the chat on the right-hand side of the screen. Hello, Oswald. Thank you for joining us.
Yeah, hello, Kate. Thank you for moderating me and the session today.
That's brilliant. So we do have quite a few questions that have come in for you, if that's okay.
Yeah, that's okay.
So should we have a little begin? So the first one's on QC, quality control. QC can be very time consuming and an expensive process. What key recommendations can be suggested to make QC processes efficient and effective?
Yes, I presented the key there is to use the consolidated quality control material. The consolidated quality control material gives you the advantage that you are dealing with less bottles, less tubes. And if you use a consolidated material, which is liquid, that has another advantage. So you don't need to spend time on reconstitution, waiting that the material is ready to use. So you have a tube which properly is already labeled, and that's the ideal way to do quality control these days. And it's available not only for clinical chemistry, it's now also available for immunodiagnostics. So a broad field can be covered by this consolidated, or I should better say highly consolidated quality control material.
Great, thank you. So are there any advantages to using highly consolidated QC material, and what are the disadvantages?
Yeah, the advantage of a consolidated quality control material is that you have less errors in the handling process. Which finally gives you a better precision or imprecision and more reliable patient results. This is also important to patient safety, because lab results are important for clinical decisions, treatments, and so forth, and monitoring, so forth on. And the other part of your question, tell me the disadvantage. That's a little bit the challenge, as I'm that much engaged with highly consolidated quality control. It's hard to say what is a disadvantage.
Yeah, okay, I see that. Thank you. That's a very good answer. For which time period should I order QC material to avoid many crossover studies?
Yeah, crossover studies are time-consuming and as well cost producing for the lab. That's something which labs usually try to avoid, and in order to do so, would be a good idea to order enough QC material, let's say for a period of 1, 2, 3 years, depends on the stability of the quality control material. Usually a quality control material is stable over 3 years, to my knowledge, even the highly consolidated QC material. And that could be a good idea to order it for this long time frame, in order also to avoid this crossover studies, which are always, as I said, time consuming and disappointing people in their daily routine work.
Speaking of QC materials, actually, we have a very good question here. Why are there two different types of QC materials, liquid and dry freezed, on the market? Can you explain shortly which is your preference and why?
Yeah, it depends where you are. So I know countries which have high temperature. Now today we have a sunny day here in Munich, and it's also quite warm. So if you have to ship the quality control material over long distance, which might be taking a lot of time and is the chance of being damaged by high temperature, then the preference is to use the dry freezed material. On the other hand, my favorite is to use a liquid quality control material, which is highly consolidated and where you have short distance for the transportation of the material. Then this is for sure a better way, as I already told everyone. That's error reduction and highly reliable material, which avoids a lot of errors. And also, if you look at sustainability, this is a new topic in laboratory medicine as well, that we have to look at sustainability. So that's more on the side of the highly consolidated liquid quality control material. I have to say there is, as you asked me, this disadvantage. Yeah, shipping a liquid material may ask for dry ice transport. And dry ice transport is also a little bit more costly than just normal mail. But again, you should have to see the balance between the cost you spend on errors, on problems, on handling time, on disappointment of everyone, the lab and the clinicians as well as the patient. So it's not that easy to answer this or that is the best.
Yeah.
You have to look where you are and what you consider. Probably cost shouldn't be that much in the focus. It should be the reliability of the lab result and the patient safety.
Yeah. Oh, that's great. Thank you.
Mm-hmm.
Can peer group data be used as an additional resource for streamlining the QC troubleshooting process, and how?
Okay, this is a question which goes further to or deeper to troubleshooting, which is, at the end, it's also part of efficiency. If you can do a proper troubleshooting, if you use the right QC material and the right analytical instrument, usually you don't have that much of troubleshooting to do. But if you have those cases, then it would be nice if the company which is providing you with the QC material is offering also a software package which taking care of peer results. Peer results are those results from all labs using the same equipment, the same reagent, the same quality control material. If you put the data all together, you get a picture of the quality around the globe. And this very often helps you to see if your neighbor labs or one of the peer group labs are suffering with a similar issue which you figured out in your lab. Then you have opportunity and good evidence to talk to the manufacturer of the instrument or the reagent, or even sometimes with the QC material, the manufacturer of the QC material, to figure out what was or what is the problem.
Yeah,
So peer group data use is very helpful, especially in troubleshooting.
Okay, great answer. So this one, do I need to take action in case a reagent lot differs from the previous one?
Yes, for sure. You have to react on this. In the past, we very often believe that lot-to-lot difference is quite small, but it could happen, especially in the field of immunoassays, that there is a large difference. And the large difference finally ends up in having issues with the interpretation of clinical situations, as the results are not the same between labs. So that's the first reaction a lab has to do. As soon as the lot changes, the reagent lot changes, you have to check if you get the same quality of results as before. If you don't, which is more or less the case these days, then you have to reevaluate your reagent a little bit and maybe changing your quality control rules according the new lot to lot difference which you are detecting. And here also sometimes the peer group values, which you can get from this software, as I mentioned before, can help you to show if there is such a difference between different reagent lots. Beside the reagent lot, you have to look also to the calibrator. Sometimes with the lot changes of the reagent, the manufacturer of the reagent change also the calibrator. So it's not that easy to just figure it out on the lot. It could be the combination calibrator and the reagent lot. If they are both new, you have to check if they are perform as the lot before. And if not, then you have to figure out the reason for it. That's mainly the difference in the manufacturing area of the producer of this reagent. You have to get in contact with the manufacturer and figure out what is the difference. Due to the new IVD, that's an in vitro diagnostic regulation here in Europe, the manufacturer is supposed to offer those details on request so that you know, yeah, with a new lot, there's a 5% difference, and then you have to react accordingly. Long answer for this.
They're thorough answers, Oswald. They're very good.
Yeah.
So we're coming up towards the last 9 minutes. So maybe we've got time for maybe 2 more questions, if that's okay.
Yeah, that's okay for me.
Okay, there's quite a few
Yes,
here.
I'm here to answer questions
today. And you're doing it very well. It's amazing. Okay, so the next question is, what should I or a lab do if QC is out of control? Keep trying the QC? Can I continue to release patient results while I troubleshoot?
Firsthand, if you're out of QC, that means out of QC regulations, rules, which are given either by the management of the lab or in some areas, some countries given by the government or other organizations. So if you're out, you're out. That means stop reporting patient results. You have to figure out what is the reason for this situation. I know sometimes people doing the measurement of the same QC material again, and again, and still hope that it will fix the problem. But the problem is somewhere else. It's not just this measurement, except if there is a mismatch of the sample of the QC material. That's why also consolidated QC material help you in getting less of those issues where you be out of control. So first, don't report patient results. Figure out what is the reason, fix the reason, and if it's done, the fixed of the reason, then do a measurement of the QC material again, see if you are in the limits. If you're still not in the limits, then it's maybe time to get in contact with the manufacturer of the reagent instrument, but also with the producer of the QC material. So I cannot say just it's there, the problem. I have to see the situation in this particular lab to figure out what went wrong. Very often, and that's what I have to mention, people are not following instructions of use. You know, I'm a man, and if I get a new device, for example, a new mobile phone, why should I read all the manual? I think I'm clever enough to just start with the mobile phone, but then I end up in having a problem. Then I remember, yes, there is an instruction for use, and that's same in the laboratory. Please carefully read the instruction for use. They help you a lot.
Yeah. Oh, brilliant. Thank you. So the next question is,
another difficult
Should I get you a very difficult one? Are there any troubleshooting actions that I should prioritize, and what are those? where can I get more information?
So as I just said, the best information you get from the instruction of use. So if you follow properly the protocol, you don't, very seldom you end up in having trouble. But if you have a trouble, okay, then the quality control is the key to detect those issues. So it's not a good idea to wait till the clinician gives you a phone call and tell you that the results you are producing in your lab is not reliable and you have an issue. So that's not the proper way to do. So you have to use a proper QC material together with some rules and regulations to figure out that you deliver a proper quality control. So quality control is the first step to see if you have a trouble. Fix the trouble. In the meantime, don't report patient results. I have to repeat this. And after you fix the problem, again, the quality control is the key to say go or not.
Oh, brilliant. Thank you so much for answering all of those questions. There's a lot more.
Yeah, I can imagine.
But we'll pause for there for now. So hang on one moment. So unfortunately, this concludes our session today, but we would like to extend our gratitude to our expert speaker, Oswald Sonntag, for his insightful presentation and his time today answering all of our audience questions. We would also like to thank LGC for underwriting today's webcast. Additionally, we would like to thank our audience for joining us and for their engaging questions. Any questions we did not have time to address today, as well as those submitted during the on-demand period, will be sent to Oswald via email after the webinar. This webcast will be available for on-demand viewing within the next few days. Lab Medica will send you an email notification when the replay is available to view. We encourage you to share the on-demand webinar with your colleagues who may have missed today's live event. And finally, thank you again, Oswald, and thank you everyone for joining us today, and goodbye for now.
Thank you, Kate. And also want to give a wave to everyone in the world, wishing you a successful day with a good quality control procedure, and hope to see you once again. Thank you. Bye-bye.
Amazing. Great final words there, Oswald. Thank you very much. Bye-bye.
Bye-bye.